Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Well, good afternoon, everybody. Welcome. And my name is Beatriz Diaz-Acosta. I work for Microsoft Research as part of the Health and Wellbeing theme. And I'm here to introduce Vince Forgetta. Forgetta. Oh, excellent. Good. Who actually, yes, who actually was our intern this summer. He comes to us from McGill University. And he did some fantastic work. I'm not going to steal any of your thunder because he's going to tell us all about it. So he's here to present BLIP, or really what it stands for is a tool to automate NCBI BLAST searches and customize the results for exploration in Live Labs Pivot. So I'll leave you to it, Vince.
Thank you for the introduction. So good afternoon, everyone. My name is Vince, and over the next short while, I'll be talking to you about the program I made during my internship at Microsoft this summer. It's called BLIP, or Blast and Pivot, and it's available at blip.codeplex.com. And after my summer internship, I'm now continuing my PhD at McGill University in the Department of Human Genetics. I hope to finish soon. Now, I'll start by explaining BLAST a little bit first. I won't explain Pivot because it's always nicer to see Pivot in action because it's very hard to explain. It's a very visual piece of software. But I will explain BLAST for those of you that are maybe not very familiar with it, but on a very basic level. So BLAST is similar to it's similar to, let's say, web searches, such as with Bing or Google, that you would input a query and you would expect a result. In this case, yes? You can't hear me quite well? Is this just record? Okay, I can talk louder. Okay, sorry. I'll try to speak louder. Is this better? Okay. And it's probably because I'm talking that way, so. No, but it's only for a recording, so. So where was I? Yes. So usually what you submit to BLAST is a biological sequence, and it returns to you information such as similarity, the species that's most similar to your sequence that you originally put in, and possibly the function of the gene if you input a gene sequence. There are two ways that you can run BLAST. You can either run it through a web service, which is available at NCBI, or you can run it locally on your own machine by downloading the program itself, or by downloading the program and also by downloading the databases themselves. Now, there are some limitations with BLAST that usually arrive when you're querying potentially thousands of sequences, such as, let's say, approximately 5,000 genes from the E. coli genome. And these limitations usually manifest themselves depending on who's analyzing the data. If it's the scientist himself who's analyzing the data and he has no programming expertise, he usually ends up either manually inspecting stuff or putting it into Excel. And that just really takes up a lot of time on his part or her part. Or he can hire a programmer to do the analysis for him, but that costs money. So that's another limitation. So that's what motivated me to make BLIP, basically, is, and this is pretty much how BLIP works in one slide with a few pictures, is you have your multiple sequences at the top, which you're submitting, potentially 5,000 sequences. You submit them to BLIP. BLIP will then, for each of those sequences, query BLAST, but the remote version, so the one at NCBI, and fetch the results as it's going along. And that takes some time, but at least it's automated, so the scientists can go away, either get a coffee, have lunch, and then eventually come back the next day and have a result. And then what the software will also do is it'll convert all that data into a collection which you can visualize in Pivot, which you can then search and see trends in his data. The software was developed using Microsoft.NET and Windows Presentation Foundation, but most importantly, it was developed using the Microsoft Biology Foundation, which for most importantly enabled me to do this within 3 months, because the converters for reading the different biological sequence formats was already done for me, step 1 pretty much, so quite a bit of it. And also step 2, for the talking between the application and NCBI BLAST, there was also an interface for that as well. So I just had to basically design the actual Windows application itself and the thing that converts the collection to Pivot and pretty much merge everything together. So luckily, at the same time that I was doing this, I was collaborating with somebody so in Canada, so at the Agricultural Food Administration, Musa Diarra, and he isolated this E. coli strain from broiler chicken, ECD227, and noticed that there were two very interesting things. The first thing was that some tests said it was E. coli, and some other tests said that it was something else related to E. coli, but not E. coli itself. And the other thing is that this particular strain is also resistant to multiple antibiotics. And the basic things that you want to try to answer out of this is, first, so what is the other species that it's indicating? And second is which genes are actually responsible for the antibiotic resistance themselves. And the one way to get at this is to sequence the genome and analyze the genome. Right? So that's what we had done. We had sequenced the genome at McGill. We ran gene prediction algorithms on it and determined that it had 5,825 genes, which we then put into Blip. You click basically next, 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 as you'll see in the demo. It's very simple. And you convert the data to a pivot collection. And as you'll also see in the demo, I'll show you how we arrived at the fact that the other species was actually E. fergusoni, which is a close cousin to E. coli, and how we can quickly search the data in Pivot and actually identify the numerous genes that are potentially responsible for the antibiotic resistance, such as pumps and transporters, which is not terribly important to know what the genes do. You just have to know how to search for them. So unless there are other questions, I will proceed to the demo. Okay. I'll try to spend more time in the demo. That's why I blew through everything. Right, so first a very short demo of Blip. Here it is. So you just have a welcome screen explaining what Blip does, some basic instructions, but you can also go to the website and there's more instructions there. I have a Word document with a full tutorial. You load your sequence files. So here I actually won't load a data set because we'll have to wait for BLAST and the downloading of all the data. I have a pre-prepared data set here, which is not the data set I'll be showing you in Pivot. It's actually a smaller data set, so I can actually run through a whole run of the program. It's actually a plasmid from E. coli. Has roughly 250 genes in it. So once we load this up. Right, so here it just prompts the user that they loaded up an existing project that already has data in it. If that wasn't the case, it would prompt you to load a FASTA file with sequences and whatnot. You can specify a name for the pivot collection and some other information. We'll leave that as default. Because everything is already submitted through BLAST, it'll just skip through this step. But here you can specify if you had submitted genes or protein sequences, whether you want to use BLAST-P or BLAST-X, the genetic code, the database, and the numerous filters. And so this basically goes through, makes sure that all the BLAST jobs are completed. If they weren't completed, it would actually complete them for you. So it's pretty neat because then, let's say you were running this for 5,000 genes and it crashed overnight, halfway through, you wouldn't have to redo the whole process. It would continue from halfway. So this will be illustrated better when I show it to you in Pivot, but one of the main things in Pivot is that each item is represented by one image. And in Blip, each image is actually a match in BLAST. So it's a match from your query sequence to something in the database. And typically, the information is all textual. There's no graphical format to this. So we decided to create something that you can create your own custom image from the data itself, from the textual data. And so you have the 22 attributes here from the BLAST report and associated records in NCBI. And here I just have a mock image layout, an example image layout. So let's say the title here is too big of the query sequence. You can reduce that down. Let's say 10 or something. The big number 1 is basically the rank of the BLAST hit. So this is the best hit. This is the name of the gene. We'll make that a little bit bigger, let's say. And you can increase the font size. right? Something like that. Okay, and so on and so forth. You can add, let's say you wanted to add the score on the bottom here. You can put that here, reduce the font. Right, something like that, put that here. Okay. And then you would click next, and it would basically create a pivot collection. And then if you had pivot installed in your machine, it will actually pop up a little button saying launch pivot, and that's it. So we won't wait for this to finish. It'll take 30 seconds. So with the data of the 4,825 genes, which I'll go to next and pivot, this is what you're presented with when you first start the program. And here we have all the individual tiny images sorted by the same order that they appeared in the original sequence file. So if you happen to, let's say, order them from the start of the genome to the end of the genome, then they would appear in the same order here. And they're color-coded by the species that they're most similar to in the BLAST database. So you can see sort of some pattern, but not terribly apparent. But just to zoom in, you can zoom in, and oopsie. Okay. I should probably look this way, it might be easier. Right, so you can see for, oops. Right, for query sequence 1, there are the 2 top hits. So for this demo, I just retained basically the 2 top hits. Hit number 1 and hit number 2, and then for query sequence 2, so on and so forth. Right? And when you click on one of the images, you get some more detail on the match itself. And when you click on links, you can get to the actual pairwise alignment that most people are used to. So if you want to copy, paste this out, or do further analysis on it. So this is usually what you get from a BLAST result, right? And then, sorry. So this is usually what you get from a BLAST result. And I just basically parse that and then visualize it in Pivot. And that'll load eventually. It takes a little while. Okay. Oh, and then the other advantage here is that you can actually click on these links, and they'll navigate to an external resource, such as the record in NCBI, the original record, and so on and so forth. So now, that was just basically how this data looks in Pivot. Let's see how it answers the 2 questions that we had before. Right? So the first question was, which 2 species is this from? So just zoom out a bit. OK. And if we order this by species instead, right? OK. And then we can plot it as a histogram. Right? And then you can also see here that it basically lists them all out, right? So you can see that the top 2 ones are E. coli and E. fergusoni, these 2 species. Right, but what I don't, so this is the first and the second hit. If let's say we would just filter it for the top 1, which we can do here, right, so that'll remove all the second hits. Okay, you can see it's sort of still the same, right? But the idea is also that in BLAST, because we're dealing with multiple hits, the first hit can actually be of the same score as the second hit. So the same similarity, exactly the same sequence in E. coli and E. fergusoni. That's not necessarily telling you which one it's from. It's just telling you it's from both, right? So you would want to limit this a little bit further by saying that the next score is actually less than this top score. So you can say with some sort of certainty that it's actually that one. So just a further level of refinement. And you can see it doesn't really change that much. And you can see that you still have mainly E. coli and E. fergusoni. The next question that you might have is, well, exactly how did this arise? Is it just, though there was, at one time, there was a half genome of E. fergusoni and it got merged with E. coli? And you should see that basically if there was some sort of contiguous block of genes that were all of the same species. So you can reorder this by input order. Am I OK for time? OK. And you can see that it's pretty haphazard, right? There's maybe a run here of something, but it's pretty haphazard, meaning that it's not one genome got integrated to another one. It's that this species is actually sort of intermediate between the 2. Right? It's sort of like a related cousin of the 2. So that's for the sorting slash somewhat filtering of the output. But the neat thing about Pivot is you can also, here, I'll just restart everything from the beginning. Remove that filter. OK. The other thing that, so the other question we had is the antibiotic resistance genes in this organism. And with that, you can just filter by keyword. So the person who I was collaborating with was actually interested in specific keywords that indicate which genes are going to be involved in resistance to drugs. And one of them, aptly named, well, so one of the keywords is actually multi-drug. So, right, so you can do that. And again, you can filter it by just the top hit. Right, and you can see that there are numerous genes that he can go through later and figure out, okay, well, it's resistant to this antibiotic because it specifically has the bicyclomycin gene. Right? And you can go through and figure out more details on that. Since I have more time, I actually have oops. Sorry, I forgot to put on my little crib notes to go over some of the other things I wanted to talk about. Oh, yes, so the other thing that's not really related to this, but is also maybe, there's this concept also in bacteriology of phages, which are basically viruses that infect bacteria, and they can insert themselves in the genome. And you can also use Pivot to find those things. You can enter in another keyword up here. Enter a new keyword, and you can look for phage. And this is nice because it shows the interplay between the filter by keyword and the positional information that you can get at the same time in Pivot. So I still got rank number 1. That's good. Import order. If I just plot the histogram, and you can see that there are some prominent clusters of genes derived from phage. Let's say something between positional or between import order 500 and 1,000. So there's a prominent peak. Between 2,000 and 2,500, you also have a prominent peak. So you can have somewhat of an idea of positional information. Not terribly accurate, but the idea is that once you have this, you can link out to maybe something else that will give you a more refined idea. So, oops, go to the acknowledgements. Okay. So firstly, I would like to thank my mentor, Simon, for having me as his intern. It was a great time. I would like to thank so 2 interns in particular that I worked with, Xin-Yi, who's right there, who helped me out a lot with C#, and we just talked back and forth on what was the best way to visualize this in Pivot, so on and so forth. And Mauro, who was the intern sitting next to me, also helped me with C#. I would bother him every once in a while. Beatrice, Vivek, Bob, and Mike, part of the MBF team. Actually got the opportunity to test this software on some users. And I'm grateful to the members of the EPX user experience group. The Azure Blast team as well just talks of actually trying to integrate this with Azure Blast so the user doesn't have to wait the 5-6 hours to actually complete all the Blast results. The Live Labs Pivot team for answering my questions. And my collaboration with Musa for the E. coli strain, and McGill University, and the sequencing platform at McGill University. Thank you.
All right. That's great. So questions. We have a few minutes for questions if anybody has some. Don't be shy. Okay, excellent.
I like it, the metaphor, the way you show each protein or each genome. Have you? I think that it will be very interesting for people that study phylogeny. This way to show because when you build the trees, the biologist he needs to identify easily, quickly, the proteins that he's studying. Do you guys have tried to do something like that instead of building a chart with many trying to build a tree?
Yeah, so by default, so Pivot only has that visualization in it, which is the bar chart or the matrix view. It doesn't have a tree view. But the Silverlight version of Pivot, which is the online-only version, actually enables plugins. So you can write your own visualization plugin for that and visualize it as a tree. But we haven't thought of it.
I have a different question, but first I'll just tell you I have a student who did that in processing, and you could probably plug it into this. But anyway, my question was, I forgot. Hang on, wait, no, I must know. Yes, okay. This looks great, and I'm just curious whether anybody has published graphics out of these displays, or whether you then are intended to export the insight that you get from this to something else that would be a more standard publication graphic.
Yeah, I guess that's maybe one of the issues with this, is that apart from taking a screenshot of it, there's nothing that you can export for publication. But also, the idea I think I had behind this software is more just as a simple tool which a biologist or a scientist can use to gain insight very quickly. But then they'll probably, so they'll eventually have to use something else later to really pin down what's happening. Right? It's something more just, I have these 4,000 genes, I need to narrow it down to, like an interesting 20 or 30. And which are those 20 or 30? They don't know beforehand, so they can use this as a search/sorting.
Can you export, in other words, you had filtered down the data set by the end of what you showed us. Can you export what you're left with in some
Right now, no. But again, so with the Silverlight version, you can write a plugin that you can basically click on one of those bars in the bar chart, and you'll have an export button or something, and you can export it. But as it is, no, it doesn't offer that functionality.
It's a very requested piece of functionality, so I'm sure the team is working on it.
I don't actually have a question for you, Vince. I just want to say something. One of the things that you mentioned phylogenetic trees. We've actually got pending incorporation into MBF. We have a set of functionality related to phylogenetics and the drawing of trees. It's one of the contributions we've had from the community. It'll be in there soon. So although that won't integrate directly with what you've seen today, what it would do is it would enable you to perhaps extend something like BLIM to include phylogenetics as well, if that's what you wanted to do. Yeah, and I was going to just reiterate what you said, which is although export doesn't currently work in Pivot, exporting a set of suitably filtered information into something like an Excel spreadsheet seems to be so commonly requested that it's going to find its way into a subsequent version.
All right. Well, thank you, Vince, again so much for the wonderful presentation. And I think that leaves us with a few more minutes, so that if you guys want to catch another talk, you can go there, or you can stay here for our next intern project, which will start in just 3-4 minutes. Thanks, Vince, again.
Thank you.
Well, good afternoon, everybody, and welcome so much on behalf of Microsoft Research in this eScience workshop. My name is Beatriz Diaz-Acosta, and I am a program manager for Microsoft Research. I work for the health and well-being theme within Microsoft Research, and I'm delighted to present today Xin-Yi Chua, who was actually an intern with us this summer and created a really wonderful project called GenoZoom. And she'll tell you a little bit more about it herself. So welcome, Xin-Yi.
Thank you very much, Bea. So thank you very much for the introduction. My name is Xin-Yi. I was very lucky enough to be a summer intern here for Microsoft External Research, and Mike Zakowski was my mentor. So I'm a PhD candidate from Queensland University of Technology in Brisbane, Australia. So without further ado, I'll just dive straight in. So the project I was looking at is called GenoZoom. It's pretty much a genome browser. So genome browsers, they provide a graphical interface for text-based genomic data or data that's stored in databases. So just a quick example of that is a common format, the GenBank format. It's all text, and you get down to your gene annotations, and if you keep scrolling, eventually, okay, they stop because the file's pretty big. You just get a whole sequence of the DNA. It's just not something pleasing to look at. So so they tried to make it graphical, as they say, a picture tells 1,000 words. So genome browsers are used in many areas of research, from sequencing to annotations to exploration to validation. And the conventional format that's used is you get your genome sequence as the reference line at the top, and it's shown as a ruler, so with your genomic coordinates. And you get different tracks, so your gene annotations following, so they can be RNA, gene features, or calculated data, which I'll show in a moment, GZig content, that sort of information. So there's actually a lot of genome browsers available out there already, and one of the key things that we didn't want to do in the project was to reinvent the wheel. But we wanted to address some of the issues that the current browsers don't do so well. So they don't scale too well. They don't provide a seamless user experience from low resolution to high resolution. So basically, I can't load in for example, entire human chromosome and go from the entire chromosome straight down to the DNA sequence in a quick and rapid time. They don't support unformatted user annotation, so if I was doing some research, I just wanted to make a note on a particular gene of interest, just for example, a post-it note, that sort of thing. The current browsers don't support this. And it's also difficult to view your own set of sequences. So a lot of the browsers out there, they have their own set of reference sequences already. So for example, the UCSC browser, which is commonly used, has about 40-odd genome sequences. But if I sequence my own data and I just want to have a look at what it looks like, it takes a while to set up the service and then put my own data on there. So that was one of the key things I wanted to address as well, to make it much easier for the user to just download and put in their own data. So the project investigation was to look at how DeepZoom and Silverlight can address these issues and make the user experience much more enjoyable. So why did I select Silverlight over a WPF version? Mainly because Silverlight was cross-platform. It's available on the web, so it makes collaboration a lot easier. It has the out-of-browser option, so you can download the application and use it on your desktop, just like a local client application, which I'll demonstrate in a few moments. And the key thing is it supports the multi-scale image control. So that's the Silverlight control to host the DeepZoom images. Unfortunately, it's not compatible with the Microsoft Biology Foundation version 1 library. So I had to develop a separate tool to generate the images, which I'll go into. The image loading depends on the network, but there is a workaround. You can host the images locally, and that should speed up the loading time. And some of the key advantages with the WPF is it does support the MPF library for WPF, but one key disadvantage was that the WPF version doesn't have the multi-scale image control, so I can't host the DeepZoom images on there. I would really like to see that supported in WPF. And without that, if I was to go with the client version, I would actually have to redo all the panning and zooming animation, as well as the data sampling algorithms, which DeepZoom just sort of provides for free or as an advantage. So just a little bit about DeepZoom, why we decided to use this as an exploration to see how it would make the genome browser much more usable is because it provides an ability to navigate large-scale data, visual data, and optimizing the bandwidth. So how it does that is it creates an image pyramid of your high-resolution data. So you start off from the bottom. You have your high-resolution image. It divides it into tiles that are 256 by 256 pixels default. And then the next zoom level up, it reduces your image by half and then divides that into tiles again. And it keeps repeating the process going up until you get a 1 pixel by 1 pixel rendition of your original image. And as you're zooming in, it uses this blurring effect and slowly crystallizes the picture. So you get a much more smoother interaction when you're navigating. And because of this way it's building the image pyramid, it essentially takes care of the data sampling for you. So I can essentially go from a very big, the entire genome, and then zoom straight down to DNA sequences. And with the ability to build different image collections, so essentially I can mix and match my own GenoZoom collection. So I can have a collection of DNA tracks, one for gene tracks, one for CG content, and then depending on the research question I want, the user can custom tailor their own GenoZoom collection, which you'll have a look at in the demo. But there was one limitation when I used DeepZoom to do the Genome Browser. And the key limitation was that DeepZoom is primarily designed for sort of rectangular type images, so ones that conform to the 4:3 or 16:9 ratios. But because we wanted to keep to a more conventional Genome Browser graphic format, which is linear, you get really thin ribbons tracks. So for example, this is the DNA sequence. It's 8 pixels high and 4 pixel per base pair. And that's for E. coli, which is about 4 million odd base pairs. So you're imagining a really long image there. And if you want to see the entire genome, when you zoom out, you pretty much get an invisible line. So that was one of the problems that I had to deal with. I tried to compensate this by increasing the height of the image, so pretty much repeating all the pixels and making a really tall image. But that had performance issues when generating the images and waste of storage. So the reason for why this didn't work, which is better demonstrated in an animation, is because in DeepZoom, what you have is, again, your high-resolution image at the bottom, which is divided into tiles. Next image on the previous zoom level reduced by half, and then the next one above. What happens is when you zoom in from your level N, it stretches the image vertically and horizontally before it replaces the tiles from the next level. So just again, stretches horizontally and vertically, and then it grabs the tiles from the next level and replaces that. But what I really wanted, the desired behavior in the Genome Browser, was to have the height of all the images at each zoom level the same. So when you zoom in, it only stretches the image in one axis and then replaces the tiles. So unfortunately, that option wasn't available in the multi-scale image control, and I tried to see if I could extend the behavior so that the zooming was only in one direction, but unfortunately, I couldn't find a way for it. So I did different trials to see what I could do for the Genome Browser. This was one failed. Can you hear me? Okay, sorry about that. So this one we have the E. coli genome, the gene density tracks. So it's like a barcode. The blues are showing genes in the forward direction, and the black stripes are showing genes in the reverse direction. So the red box is showing what would happen if I zoomed in with my mouse wheel, and eventually you see more tracks popping up. So that's the GC content. Keep going in, and then you see gene tracks coming up. keep going in, eventually you'll get down to the DNA sequence. But the problem with this one is, what happens if I want to see the GC content next to the DNA sequence? Because I've zoomed in so far, I lose context of where I am, and I can't have the tracks next to each other. So unfortunately, that didn't work. Other attempts was to manually go into the zoom levels and actually tweak the image tiles and have different heights to try and get that navigation working. But unfortunately, using this way, you get this popping effect, so you can see sort of a transparency from the layer beneath. So that itself didn't work out very well, because you get this popping effect, and it's just not nice. So what I then came across was a concept called dynamic deep zoom, where what usually happens with the multi-scale image controllers, it sends HTTP requests to your server, and that comes back with your image tiles, and it lays it out on the screen. So with the dynamic deep zoom, you intercept this request, generate a handler that actually generates the image tiles on the fly, so you can calculate all the images on the fly and then send those back to the multi-scale control. So that was a possibility. The most common example that they have is the Mandelbrot plot, which I tried out, and it's pretty quick. When you zoom, the response time is quite reasonable. Unfortunately, I couldn't go further with this solution because of the internship time, and this one needed a database backend to support all the genomic data. And because I had to weigh up how much time left, I had to put this on hold. So in the end, we came up with a 3-component model. And I will demo this, which is probably easier. So the 3-component model, it's showing the E. coli genome. And we've got the navigation view, which shows the entire E. coli gene density tracks. The region component, which has a more zoomed in level up to 25,000 base pairs, and a details view, which goes up to about 400 base pairs. So the user interaction happens in these two, the region component and the details component.
I think this is right, because I think I know this, but I'm not sure anyone else knows.
Okay.
The little red region on the top one indicates...
Yes.
The little red region in the middle.
Yes. I was just about to say that. So that, as Simon pointed out, that red window here corresponds to the region component, and the red window in the region component corresponds to what's in view in the details component. So users can zoom in and out. And as you zoom in, because your location has moved, it will correspond in the next view. I have to just point out that I did test this before. It's just the net connection is slightly slow, so you will get this sort of frozen effect. But I'll just continue. So my browser, my apologies. Try to avoid moving my mouse up there. Okay, sorry, I'll just go on. So with this, you can search genes. If I was looking for sort of toxin-related genes, it'll jump to the correct location. And I can make unformatted user annotations in the form of push pins, so tags. Change the color as you like. reordered tracks around. And I can keep going down until I get the DNA content. And what's more is because they're all images, if you have the DeepZoom image location either hosted locally on your machine or on a web server, you can just refresh this so we get the middle section. You can add in your own custom tracks. Just need the address. So you can turn tracks on and off, add new ones. My apologies, please bear with me with the time. But there was another problem I noticed with basically when you had graphs that have peaks and troughs. When you zoomed in, because of the track, limitation of the track height, you notice that when you keep zooming in, the peaks of the graphs will clip off. So I was looking at different types of visualizations that could deal with that. And in this track, it's supposed to be showing a heat map that takes up the entire height. So it's something like this, but it takes up the entire height where the color tells you the value. So I was testing out with these different types of images. But eventually, basically that just shows you that because they're all images, the user can essentially put in any image they like as long as they've got a genomic location and then put it up as a track. So that was the idea there. And then you can download this with the out-of-browser facility available in Silverlight. And when I open that up, you'll have, or when it opens up itself, the exact same user interface as the one that I was demoing. So that, and if you had your own genome sequence, convert that into a DeepZoom image collection, then you can just see your own image data straight away without the lengthy install process. So, as I mentioned before with Silverlight, unfortunately, it's not compatible with Microsoft Biology Foundation version 1. So I had to build the GenoZoom image generator, or the GZig tool, as a separate control. So the input file is a GenBank file. For the prototype, that's it supports the GenBank and FASTA formats. Uses the MBF parser, creates the raw images, which you put into the DeepZoom tool to make the DeepZoom image collection, and then that's what you see in the GenoZoom, so the different tracks there. There is a known issue with this process, is that the image generation can take a while, but if you're dealing with reference data, it's not too bad because you probably only process that once in a while and you use it over and over again. And also to address the image generation issue was what I mentioned before, was the dynamic DeepZoom, where you generate the images on the fly. That could also address the problem. So some disadvantages of the GenoZoom application is the user can't dynamically change the visuals. For example, if I wanted to highlight all regions where the GC content is above 0.5, for example, make them red. Unfortunately, because they're all static images in these processes, I can't do that. Custom data has to be hosted by a web server. So again, that is, if the multi-scale image control is available in the WPF, maybe I could do more a local version. But at the moment, that's not possible. Data size explosion from text to images, there is a noticeable issue in that. But with images, you can encode much more information. So for example, you can color code your genes based on functionality. So you can do a lot more with images and include more data in there. And again, the dynamic deep zoom approach where you generate the images on fly could also address this issue. The performance, as we noticed today, unfortunately, is limited by the network, but if the images are hosted on the local machine, that would be faster. And just minor sort of known issues and bugs is the animation. If you are too quick on the animation before one finishes and you move, it does have a little bit of unexpected behavior. And because I was trying to have different ways of interacting with the data, so the user can move the red window or they can move the actual component itself, there is sort of a cyclic dependency where the input's coming from that has a slight behavior. Just needs a bit more testing in that one. But the advantages is that with the deep zoom approach, I have much smoother interaction, so I can go out from an entire genome and down straight to the DNA level. It would work much better if I had just a one-dimension stretch instead of the two-dimension, as I mentioned. But I have the higher data resolution, and I can support unformatted user annotations with the push pins, so they don't have to be in fast format or that sort of thing. It's easier for the user to view their own data, so as I mentioned, you can download the tool, or if it's on the web, as long as you've got your address to your own genome collection, you can just put that in and then it'll pop up. Easy to set up. Again, with the whole different tracks, you can build your own GenoZoom collection just to support your research questions. And because the images, the user can essentially generate any image that they can think of. Future work, exploration with the dynamic deep zoom. I really want to try that out and see how far I can take with that. It was one of the long-term goals was actually to integrate this with Pivot. So Pivot exploration, as Vince demoed before, was what happens if you come to a collection, you want to see where do all these genes appear on your genome. So we would export that collection into the Genome Browser and have them appear as the tags, and then vice versa, export your user annotations into Pivot, and then you can do further analysis that way. So unfortunately, again, due to time constraints, we didn't get that far, but it's in the future work list. Link out to external data sources, click on a gene, you get more information about the gene or the PubMed or literature publications. I've mentioned the tailor build your own genome zoom collection, changing the zoom behavior to just one-dimensional stretch, expand the GZig tool support just to incorporate more input file formats. And of course, it would be nice to compare this tool to a pure Silverlight or WPF version just to see how performance would pan out. But just to keep in mind, if I was to do a pure client version, I do have to redo all the animations as well as the data sampling, which is a non-trivial task. Availability, it's in the process. It will be available on Coplex under the MSPL license. Just having some paperwork at the moment. So visit the MBF website for the release date. And thank you again to Mike, my intern, and MSR External Research. Sorry, my mentor, not my intern. I'm the intern. My apologies. Sorry. My mentor and the external research team. It was really fun working with everyone there, and they were all very friendly and welcoming and eager to help. Vince as well for all the talks back and forth. It really helped out. And just a lot of people, which it's always dangerous. People, you might leave people out. But thank you, everyone, and thank you as well for coming to and listening to my talk. So that concludes this. Any questions?
That's marvelous, Xin-Yi. So now we can take some questions. Now I must warn you that we are using this mic that you can't hear for the purpose of the video, so I will beg you to please speak into the mic when you ask your questions. I'm going to take him first. I'll come back over here.
Thanks. One very obvious question, I guess, one that we've experienced as well with previous work. You've done something there with E. coli.
Yes.
Did you try it with a much larger chromosome? And if you did, what were the experiences that you had?
I should actually have explained that. The initial drive for the project was to try and do a human genome browser. But two things came up with that. First, it was because we had that long-term goal to interact with Pivot, and the data case with Vince's project was E. coli. So I chose to do the E. coli tracks first. Second one was pretty much image generation, the performance, and the storage. For human, it would have been too long and too big, and just intern time sort of didn't pan out. So unfortunately, I couldn't try that, so I'm not too sure what it would have been like. But theoretically, I think it would be pretty much the same, because the images are all generated and on the server anyway.
Yeah. We have done some very, very brief experiments and prototypes with the human genome, but focusing from chromosome to gene level, not going all the way down to base pairs, which would actually, of course, cause the data explosion, but it could be possible. It's just, we just need Xin-Yi to come back and do it. Thank you.
Hi. I was actually wondering about the dynamic zoom thing. So I was wondering how far did you actually go on success level? Like, were you able to create some images, like certain region size, like 500,000 or something like that?
Unfortunately, I couldn't. The only extent was I tried out the Mandelbrot plot, because when I read about the dynamic deep zoom, one key problem that I was really concerned about was the response time, because you're creating all these images on the fly and you're doing so many tracks, whether it would be reasonable for the user to sit there and wait. So that was one problem that I was really worried about, and that's one thing I want to actually test out. Unfortunately, due to time constraints, I couldn't test it out. I could only look at the Mandelbrot plot example, and that response time was quite reasonable. I can't remember the URL. If I could, I would show you what it would be like. I'm happy to try and find out after the talk with you. Yes, unfortunately, I'm stuck to answer that question because I didn't have time to try it out.
The pre-processed images that pre-created images, are this out of some reads or?
No, those one was from the reference E. coli GenBank file. Yeah.
Another question over here.
So it seems like you've derived some pretty good requirements for a one-dimensional deep zoom, right? And it seems like there's probably other applications that would benefit from that, you know, big time series or something.
Yes,
is that a next step that you'd like to maybe work on? Is what would a one-dimensional deep zoom look like, or is anyone else working on that?
Yes, that's a really good point. The one-dimensional deep zoom is not just applicable. This is just one example. I think there's so many other examples that can fit with that. I would love to be able to work more about that. I thought it wouldn't be too hard with the multi-scale control because all the behavior is actually in there. If we could separate, because what's happening is in that image control, it's sort of married the horizontal and the vertical into an aspect ratio. If we could sort of separate that out into just a vertical and a horizontal component, I think it's not too hard to work out. I'm not sure if I answered that question, but
we'll definitely forward the request.
So I just want to exemplify Xin-Yi's attempts here, because when we ran into these problems and all the problems that she identified, to me that was as much value in this research as where we ended up in identifying things like this 1D zoom, which once we figured out kind of where the issues were and got connected with the right people, it was like, oh yeah. So definitely doable, and I think, you know, as we go down the road of investing more in this, we're going to be looking at that. The other thing on Well, I just came out of another talk where I'm talking to your guys, the other thing on the large data sets, the human, which was our target, the pre-processing is, you didn't really go into how much time that took, but it was pretty heavyweight going from the text-based files to these images. I mean, there's a lot of pre-processing steps there. So how long it would take to generate the human genome images is the big question. Once they're generated and you have the reference, storing that remotely combined with the dynamic deep zoom of sequences of interest, really that's the scenario that I really think starts gelling in my head about where this could really come together. And I'm not sure it also came across either, but the original idea was these image pyramids doing semantic zoom, so that at each level of these pyramids you're actually bringing in new and different information. So you might see the genome and the barcode chart, and as you're zooming in, you'll discover other information that's there. So so these are all the concepts that Xin-Yi really.
I should point out, with Mike mentioned the semantic zoom. I did actually get that working in the beginning. It just didn't make its way into the end result. So I did have. You start off with your genome, you zoom in, sort of like maps. When you get the country, you zoom in, you get states, and down to the street level. So you get more information coming out. So I did get that working, but just didn't make it. And just one fun comment about the one-dimensional zoom. Someone actually commented another use case for that would be comic strips. So there you go.
Yes.
Any other questions?
And for comic strips, do a search on Infinite Canvas, and you'll see some cool experiments.
I just have a quick question. I thought that was fantastic, and since I don't look at gene sequences very often, I'm just curious, people who do, did they get to see this tool and compare it to other ones? And how useful was
I
it?
did manage to give a really quick demo after the internship to a Associate Professor Tim Reed in Emory. And yeah, his response was really positive about it. He pretty much just thought, oh, that's just like maps. And then, yeah, very positive about it. I'm sort of waiting for it to be released on Coplex and then making people use it and developing it more as well.
Right, one more question if we have. All right.
So you had a slide on the comparison between WPF and the
Yes.
Silverlight version. I was wondering if there is actually a version of Pivot that would integrate into a WPF.
Sorry, I can't hear you quite well.
I was wondering if there's a version of Pivot that would integrate into a WPF application. Pivot, but with DeepZoom.
Okay.
Because I was only aware that Pivot runs under Silverlight.
Oh no, the original Pivot is an actual WPF application.
Oh, okay.
There's two versions. One is a WPF application, and then the second version that came afterward is a Silverlight.
Okay.
You can have both.
Okay, that's good to know.
Sorry, I ended up answering that question. I used to work for that team.
You're the right person to answer it. So I think we have one more question up
Oh,
front.
excellent,
I
Donald.
just wanted to make a comment too that DeepZoom, there's a open source Ajax implementation of DeepZoom as well that the Maryland Institute of Technology and Humanities just released some wonderful blog posts talking about how to use. So there's a lot of ways to approach this problem if you're thinking about HTML5 or things like that. So.
Yeah.
Well, thank you, Xin-Yi, again so much for the wonderful presentation, and thank you all
very
for
much.
attending today. And with that, we'll wrap it up and call it a day. Thank you so much, everybody. Thanks.